Hello class, it's time now for a lecture from chapter 49 on somatosensory function, pain, and headache. The majority of this lecture is going to focus on pain, and then I will follow up with some different types of reasons for headache, which I would argue is still a continuation of speaking about pain. In some cases, when a person is suddenly exposed to a painful stimulus, they don't necessarily feel it right away. I have an example of that. It actually happened to me. One time when I was a teenager, I was babysitting for a very large family, had lots of kids, and these kids would eat an exorbitant amount of food. The 2-year-old would eat 2 boxes of mac and cheese, Kraft mac and cheese, on his own. It was absolutely disgusting, the amount of food I would make for these kids. Anyway, the parents would inevitably trick me into thinking they would have the kids fed and then go out. Well, something would always happen, and they'd call me early, and I would end up making food for these kids. Well, I was making, I forget, a lasagna or something like that. It was one from the freezer. It wasn't like I was making it from scratch. But I remember pulling this casserole out of the oven, and the broiler was on. It was meant to crisp the top of this casserole. And as I reached in with the pot holders to grab the both ends of this casserole dish, I straightened myself up before I had completely pulled my hands out from inside the oven. And as I did that, the top part of my thumb, not my thumb itself, but the actual metatarsal, that attaches to your tarsals, not metatarsals, metacarpal that attaches to the carpals. Anyway, just that fleshy, meaty part of the lower base of your thumb, mine hit the broiler itself. And I remember instantly feeling a sharp pricking kind of pain, but then nothing. And I stood there for microseconds, but I was completely conscious of this. And I actually remember having the thought, well, this isn't hurting. And I remember watching my skin melt. And I remember thinking in my head, this is so cool, but this should hurt. And then the pain came, and I pulled my hand away, and my skin stuck to the broiler and was just flapping in the thermal breeze. And there was a throbbing, intense pain that followed after that, which brings me to mind, this is everything to do with my talk with you today. The first contact I had with the broiler, it wasn't pain per se, but I could feel, like I said, a pricking or a stinging feeling, but it wasn't really pain. And then it was followed by that burning sensation, that this throbbing sensation when I withdrew my hand and after. And that's what pain is. It's really a twofold modality. You have a fast pain, and you have a slow pain. The fast pain is called your neo pain, and your slow pain is called your paleo pain, because we really see it from an evolutionary point of view. There are 2 different pain tracts in the spinal cord. A tract, remember, is a collection of axons inside the central nervous system. Now, this is going back to your anatomy, folks, and it's going to get technical, and I will do my best to review the anatomy of the spinal cord, but pals, I hope you still have an anatomy book where you can look a lot of this up if you get too lost. Okay, so there are 2 different tracks, and there are 2 different pain tracks. The fast pain is a newer track in evolutionary terms, and we'll differentiate that anatomically, what that is, and then we'll also address the slow pain, which is more of an ancient tract or older pain response. So we always talk about it in 2 different conditions, and we'll come back and talk about it in some detail. Again, the newer on the evolutionary scale is the fast track or the neo tract, and the slower, more burning, throbbing pain sensation is the paleo or older tract. Okay, so as I said online, I would rather err on the side of being somewhat conservative and review a little bit of the anatomy. Now, up here you're reading functional classes of neurons. Functional meaning what are the terms we use to describe what kind of action potential a neuron is sending, or in which direction is it sending it is probably the better way of explaining this. So whenever I use the word sensory, this means it's incoming information. Information from your skin, from your environment, being detected by nerves or receptors, and relaying that information back to the brain or spinal cord. Another way of saying this is afferent. Now it's really pronounced afferent, but for the sake of online learning, I want to make sure you hear me enunciate. So I'm going to purposely pronounce that word in the incorrect way. So anything that's incoming, relaying information about what's happening to the body, is sensory or afferent. It always begins, that kind of neuron will always have its nerve endings, at the beginnings of it, if you will, out in the periphery of the body. And the terminal bouton will always end in the brain and spinal cord. That's where it's going to form synapse. That's where it's going to release neurotransmitter and perpetuate this signal into interneurons. Now, interneurons are neurons that are fully contained in the central nervous system. Their soma, their dendrites, their axon, their terminal bouton, all of it, the whole package, totally contained within the central nervous system. Most of the neurons in your body are of the interneuron type, completely contained within the CNS. Now, a motor or efferent, which is really pronounced efferent, motor neurons are going to always begin in the central nervous system, but end or terminate where their terminal bouton is, out in the periphery. And these will always target some sort of effector cell, a cell that will respond to that neuron. Examples of effector cells would be skeletal muscle. That's considered to be a voluntary effector cell because you have conscious control over it. But there are involuntary effector cells as well. Smooth muscle, cardiac muscle, glands, even adipose tissue is now considered to be an effector cell type. So, look at the bottom of this slide and notice that we are really playing with a reflex arc. In the beginning, you're seeing the woman with receptors in her skin, say temperature receptors. These temperature receptors are going to relay the information about the temperature her skin is being exposed to. Maybe it's the broiler in the oven. She opens the door and that big waft of air, hot air, hits her on the skin. This is going to send action potentials through the peripheral process of that sensory or afferent neuron. The cell body for an afferent sensory neuron is always found in the dorsal root ganglion, which is listed here as the posterior root ganglion. I'm going to say the DRG, dorsal root ganglion. So you're seeing that this neuron has a peripheral process that's bringing information from her skin, but also a central process. And the central process is the part from the neuronal cell body to the part that terminates on that purple interneuron. Now, a lot of physiologists and anatomists argue over the peripheral processes and central processes and saying, are they dendrites or are they axons? And the physiologists will always say they're axons because they have voltage-gated channels on them. The anatomists will say, nope, 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 that's not going to cut it. So that's why we just call it a draw, and we refer to them as the peripheral and central process. So the peripheral process, if you wanted to label that on your slide, I would make a line to that orange axon where it says afferent input transmission. That region from the beginning of her skin receptors to where you see the cell body of sensory neuron, that whole span is the peripheral process. And then from the cell body in the dorsal root ganglion to where it terminates on the interneuron, that's the central process. And then the interneuron is where we're going to have integration. That's an important part of an action potential because your brain and spinal cord need to collect this incoming information, integrate it, and by integration I mean they need to process it, figure out what's best to do in response. And then the motor response, the efferent, the outgoing command, the action potential that will ultimately reach the effector needs to be transmitted through a motor neuron. Now, motor neurons can be further divided into somatic motor neurons or visceral motor neurons. Somatic motor neurons will terminate on skeletal muscle, as shown here in this picture. Visceral motor neurons will terminate on your involuntary effectors, smooth muscle, cardiac muscle, glands. You have no conscious control over those, even adipose tissue, as I said before. I think it's a very good idea to continue with another slide to review. And in this picture, you're seeing the 5 components of a reflex arc labeled for you. In step 1, we have to have a receptor. Now in this picture, you're seeing a nail being driven through a person's skin. Obviously, this type of stimulation would be detected by a special kind of receptor called a nociceptor, which is a special way of saying a pain receptor. And the nerve ending is going to detect this stimulus and generate an action potential that will travel through a spinal nerve. So if you're following the blue axon, you're going to see a little black swirly curvy arrow leading to the spinal nerve, where it says number 2, nerve impulse travels through sensory neuron to the spinal cord. If you travel to the dorsal root ganglion, that little expanded region where you see a tiny little blue circle, that's the dorsal root ganglion. Now, just to remind you, we have all afferent incoming action potentials need to come into the spinal cord through the dorsal root. And in step 3, you're seeing that sensory afferent neuron terminating, synapsing with the purple interneuron. And the purple interneuron cell body is found in the dorsal gray horn. The gray matter, the spinal cord, kind of looks like a butterfly, don't you think? And the part of the gray matter that's on the back side is called the dorsal gray horns. And the part that's on the anterior side, where you're seeing that orange circle for the motor neuron, that's called the ventral gray horn. Now, the part that's going across, connecting the 2 wings, where you see the central canal that would be filled with cerebral spinal fluid, that's called the central commissure. It's basically interneurons communicating from one side to the opposite side. And that's really important. Notice that this reflex arc is all ipsilateral, meaning same side. Let's say we're dealing with the right side of the body. The right index finger was pricked with the nail, and the output command is going to tell the right biceps brachii to contract, and then we withdraw our hand from that nail. That's a withdrawal reflex. That's an ipsilateral, meaning same side stimulus, same side response. But we're going to see today that the majority of these reflex arcs that I'm going to be telling you about are going to be contralateral, meaning there's going to be crossing over. And this is particularly important because the right side of the brain controls the left side of the body, and the left side of the brain controls the right side of the body. And when we're talking about sensory incoming information, the right side of your brain is processing information, sensory information, that comes from the left side of your body. And your left side of your brain is processing information about stimuli that are affecting the right side of your body. So, we are going to primarily talk about only half of these reflex arcs. Our primary focus is going to be on the sensory afferent incoming action potentials. Coming from the periphery of the body, the skin. Are we touching a candle flame? Are we poking ourselves with a tack? What sort of sensory modality are we talking about? We have to get it to the spinal cord, and then the next part we have to get it up to the brain, where the brain is going to process this information. You are going to, and we're going to deal with pain specifically. I will address touch, light touch, discriminatory touch. Obviously that's not painful. I'll tell you about those incoming pathways, because obviously you can touch something and you know the difference between a pencil in your hand and a pen in your hand versus a piece of chalk in your hand without even opening your eyes. So I'll talk about discriminative touch and the ascending pathways. But then the primary focus is going to be on the ascending pathways, delivering information about pain. That's our biggest focus. So let's go back a little bit, and we need to think of two primary different pathways for sensation. Now, I didn't say pain, I just said two different pathways for sensation, anatomical pathways. We think first of all the lower portion of the body, the neck down, and then we think from the neck up. The body, of course, is divided in many ways. That's just one way of dividing it from an anatomical view. From the neck down, we're going to have first ordered neuronal cell bodies, and the cell bodies are going to be found in the dorsal root ganglion. And remember, a dorsal root ganglion is a collection of neuronal cell bodies found outside of the central nervous system. So, the blue axon you're seeing in this screen on slide 3, and the little blue cell that's in the dorsal root ganglion, that would be a first ordered neuron. It's the first neuron relaying information. And this first ordered neuron, of course, is traveling through a spinal nerve. And how many spinal nerves do we have? We've got 31. And they go from the top of the neck all the way down to roughly the level of L2, where the spinal cord ends. And then in the vertebral canal, of course, the rest of the nerves travel as the cauda equina, which means horse's tail, until they get to their appropriate level, through which they will exit through the intervertebral foramen. Now, what about from your neck up? Obviously, if I were to drive a pencil through your cheek, you're going to feel that, right? But it's not a spinal nerve that's going to relay that information. From the neck up, now we're talking about cranial nerve number 5, the trigeminal nerve. So for our purpose today, we're going to be addressing from the neck down. We're going to be talking about afferent nerve fibers coming through a spinal nerve and then terminating on the dorsal gray horn on an interneuron. That first blue fiber that's relaying information about the stimulus, like I said, is called a first ordered neuron. And in order to relay the information up to the brain so that you can figure out, number one, firstly, what just happened to me? Is this painful or not? B, number two, secondly, if it's painful, where in the heck did this just happen to us? Because obviously, your brain, if you were talking about a painful stimulus, needs to be able to detect where this is happening in the body, and secondly, how intense is this pain? It has to give it some sort of rating system. Because obviously, if something is particularly hurtful to you, and you're not able to figure out where the hurt is coming from, then you can't remove that body part from it. And pain is a protective thing, students. It ensures that we don't just keep doing whatever we're doing. I mean, it's very aberrant for you to be, let's say, rubbing your wrist across a serrated knife. That's not what we do. We don't keep saying, ooh, this feels so good, I think I'll take another pass. The idea is that pain protects us from something that can do great bodily damage. All right, so let's move on. I hope that was a good review for you. Again, we're going to be focusing with the 31 spinal nerves for that first ordered neuron. We will not address the trigeminal nerve because the circuitry going from the face to the part of the brain for interpretation of pain is slightly different. Not hugely different, but it's just easier to get the concept using spinal nerves. I'm not cheating you, I promise. Trust me. Okay, so sensory modalities, meaning besides hearing, seeing, tasting, smelling, what other types of sensory forms are we capable of? What are we capable of detecting? Well, when it comes to our fingertips, let's start there. We can detect touch. We can detect touch and determine a shape of a structure, texture of a structure, and the specific receptor that allows or picks up on that is something called a Meissner's corpuscle. And here, just to remind you of your integumentary system, Meissner's corpuscles are found very close to the surface of the skin, and they're found in the dermal papilla. Now, shown down at the bottom of the screen are Merkel discs, and they are also found in the superior region of the dermis, and those are going to be able to detect fine touch and relay the information back to the central nervous system. There are also free nerve endings that are found in the dermis, and these are capable of detecting temperature, and they're capable of detecting pain. Now, let's talk about temperature, because 45 degrees centigrade is about the threshold. Now, your body temperature is 37 degrees centigrade. Now, usually a 10-degree shift in centigrade temperature, greater or lesser of 37 degrees, is usually detected as a temperature change. So as you drop from 37 to 30, your brain would interpret that change as, ooh, we're getting cold. As you shift from 37, say, to 40, your brain would interpret that as, ooh, I'm getting warm. I can do the same thing like taking, let's say, a piece of metal and heating it up very slightly in a flame and then putting it on the belly of your forearm. And as long as it's below 45 degrees, you would say, oh, that feels warm. But if I were to overshoot that, that's the threshold for pain. Most people who are studied would then say this is no longer a warmth issue, this is a painful stimulus. So what I want you to hear is that free nerve endings can kind of blend temperatures and pain. And I also want to share with you, my husband, we have a pool, and that man will not swim in our swimming pool unless it's at least 90 degrees. Now, even if it's a nice 80 degrees and he jumps in, when he comes to the surface of the water, every single time he'll say, ow. And for him, his free nerve endings are more sensitive to slight changes in temperature. So something that he perceives to be too cold, for most normal people, it would just be, this is a little on the chilly side. But for him, it's painful. He really is in pain. So there's a fine line between temperature and nociceptors, which means pain receptors. Again, free nerve endings, they're going to detect both of these. Then, of course, we have Pacinian corpuscle. I just love saying that. I think that's fun. And Ruffini organs. Now, both of these are going to detect pressure and vibration in the case of the Pacinian corpuscle. So, fine pressure, vibration versus deep pressure. Now, even deep pressure, if I were to squeeze really hard on your thighs, eventually that deep pressure will shift over to, this is no longer just you pushing on me, this hurts now. So if you're reading your textbook, they actually refer to these overlapping of receptors generating action potentials as the pattern theory. Whereas what that basically means is, to a certain extent, all of these receptors are going to respond to a certain stimulus, temperature, pressure, etc. But if the stimulus gets really great, all bets are off. Those free nerve endings are going to respond with those other receptors, and your brain is going to now say, this is no longer fun. This stimulus started out okay, but now it's just outright painful. Okay, so I hope you're having fun with this so far. Okay, so I would like to go over a few General ideas and terms. Okay, so first of all, we're talking about pathways. That's really what we're talking about today. And when we're talking about pain and pain pathways, we're talking about some sort of stimulus happening to your body and the relay of action potentials up to the brain for further processing. So your brain can figure out, like I said, where is this happening? How intense is this? And what's the best appropriate response? And we're not going to talk about the outgoing command. We're only worried about the information getting to the brain. Now stop and think. I'm sure you can appreciate that there's a heck of a lot of difference in pain and bleeding in a paper cut versus if you accidentally cut off your index finger with scissors. Yes, both are painful, but one leads to mild shouting of expletives, whereas the other one might lead to you passing out, right? One you will probably wash and put a Band-Aid on. The other one you should seek medical attention. So your brain needs to figure out when you've cut yourself, given yourself a paper cut versus cut off your finger. So some terms. When we're talking about a pathway, we're talking about a tract and a nucleus. Tracts are groups or bundles of axons that travel together within the central nervous system. Each tract may work with multiple nuclei. A nucleus is a collection of neuronal cell bodies located within the central nervous system. So if you look at this picture, you see the part where it looks like a thickened blue tube surrounding that blue secondary neuron? That's a tract. That's a collection of axons. Now in the picture they're just showing you one inside this tube, but they could have colored, 100 of them. That's a tract, a collection of axons. Now where is the cell body? Now you'll notice that the blue axon terminates up in the brain on a green neuronal cell body. And you'll notice that in this picture it's kind of colored a brown color. That's representing gray matter. Anytime you see gray matter inside the central nervous system, the first thing that should come to your mind is, I'm dealing with neuronal cell bodies. Why? Well, neuronal cell bodies don't have myelin on them. Otherwise, how would they respond to neurotransmitters? So most of the time when you see gray matter, you should be thinking to yourself, those are where cell bodies are. Nervous system pathways are sensory or motor. Now again today we're dealing with sensory pathways, and these are also called ascending pathways because the sensory information gathered by the sensory receptors ascends through the spinal cord to the brain. Meanwhile, motor pathways would be called descending pathways because they transmit motor information from the brain down through the spinal cord to effector cells like muscle or glands. Now again, we're going to be talking about ascending pathways, sensory, afferent, incoming. Most of our sensory pathways I'm talking about today, in fact I'm talking about the big two, and they do have decussation. Decussation means to make the form of a cross, to cross over from one side of the body to the other. The crossover process, called decussation, means that the left side of the brain processes information from the right side of the body, and vice versa. I told you that. The term contralateral is used to mean the opposite side, whereas the term ipsilateral means the same side. And of the pathways we're talking about today, the great majority of them will be crossing over. And listen, where they cross over will be different for the two pathways I'm going to tell you about. Some will stay ipsilateral for quite some time, get close up to the brain, and then cross over. Some will cross over immediately in the spinal cord and then travel up to the parts of the brain. Alright, I hope you don't have any questions with that. On the next slide, I'm going to continue on with some terminology. Okay. In most pathways, there is a precise correspondence of receptors in body regions through axons to specific functional areas in the brain. This is called somatotopy, which means body place. And I'm going to show you a picture of a homunculus map. And basically it shows the surface of the post-central gyrus. The post-central gyrus is the somatosensory cortex. And I'm going to explain to you a series of experiments. But basically, a scientist, specifically Wilder Penfield in the 1930s, he basically, on patients who were having brain surgery, anesthetized their meninges, took an electrode, and all along the somatosensory cortex, the post-central gyrus found on the parietal lobe, he kind of randomly zapped them. And he would say to them, "How are you feeling? Are you feeling any pain?" And what he thought was interesting was, you know, depending on where he was zapping the electrode, let's say he was on the right side of the brain, the patient might say something like, "Well, I'm not experiencing any pain. What you're doing is fine to me, but could you please take my left hand out of the ice water?" So by zapping all along this certain part of the brain, he was able to map what part of the body that region of the brain was interpreting sensory information for. And basically we use that map and we describe it as a homunculus, a little person drawn on the surface of the brain. Of course there isn't a little person on your brain, it's just a map. As I said, all pathways are going to have tracks and they're going to be paired. So again, we're going to have tracks that are linking the right side of your body with the left side of your brain. And we're going to have ascending tracks linking the left side of your body with the right side of your brain. The pathways that I'm going to talk about today are going to be composed of 3 neuron chains. There are 3 usual ascending tracks that are generally discussed in an anatomy class. One of those 3 has a 2-neuron chain, and that's the spinocerebellar tract or pathway. I'm not going to discuss that today. I'm talking about discriminative touch and pain. And those pathways use a 3-neuron chain. Now, these ascending pathways are named for their origin and destination. For example, one of the pathways I'm going to tell you about today is the spinothalamic pathway, which means the information is relayed from a neuron that the first neuron will end in the spinal cord, terminate there, and then the second and third neuron are relaying the information up to the brain. So we're going to start from the spinal cord and end in the thalamus. That's basically how they're named. Okay, so I want to first review with you these pictures. Now, I want you to look at the left side picture, and you're seeing a part of the brain colored in blue. And in the same picture, but towards the right, you're seeing a different region of the brain, but colored in kind of a peach red color. Now, focus on the peach red color. And what you're seeing is the post-central gyrus found in the parietal lobe. If you look on the far right picture, where it has a picture of the brain colored pinks and dark blue and aqua blue, this peach part that I'm going to discuss is shown on the right brain, right-handed picture, the picture of the brain, as the dark blue region. So that's the post-central gyrus. And on the post-central gyrus is the part of the brain whose job it is to interpret touch, pain, pressure, and temperature. So your right post-central gyrus is called the primary somatosensory cortex, and it would interpret these sensations from the left side of your body. Your left primary somatosensory cortex, found on your left post-central gyrus, would interpret pain, touch, temperature, and pressure from the right side of your body. Now, you'll notice that on that little peach-colored portion, you're seeing a picture of body parts. You're seeing a large hand, a large face, a big tongue. And this was that Wilder Penfield experiment where he took an electrode all along the length of that dark blue region shown on the right-hand picture of the brain, and he just randomly went along the entire length of it and zapped these people. Just a gentle little electrical stimulus, nothing too much to fry the brain. But anyway, so he zaps along and he's asking the patient, how do you feel? How do you feel? How do you feel? What are you feeling? And the person would describe, I'm feeling a tingling sensation on my lips. And so then he knew that region of the postcentral gyrus was responsible for interpreting sensation of the lips. And the larger the picture that's shown here, the more neurons dedicated for sensory interpretation for that body part. Now he did the same thing for the primary motor cortex, which I won't talk too much about today. But you still can do somatotopy, which means the larger the body picture, the more neurons dedicated to that spot. Now, think about how sensitive your face is, how sensitive your hand is, and look how large the pictures are drawn for the primary somatosensory cortex. Look how large the tongue is. And then somewhere on there you can find the genitalia somewhere down at the bottom below the tongue, which is kind of weird to think about for its placement. But, yeah. Oh, no, they're not below the tongue. They're on the opposite side. They're below this little homunculus guy's feet. You can see the little term that says genitals there. Now look how small they are. And just for a moment, think about how sensitive your genitals are. And I would argue it's a good thing that they're drawn so small on this picture, because if they were drawn the size of the face on this picture, I don't think we'd ever leave the house. We'd be grinding up against furniture all day long because we'd be oversensitized. Anyway, sorry, it's a little joke. So to get back to the sensory cortex, this is found on the parietal lobe, like I said. And I just want to talk about some other association areas, some interpretation areas. There are other areas found around the primary somatosensory cortex that help you interpret objects even when your eyes are closed. So if I were to, like I said, put a piece of chalk in your hand, you would probably be able to interpret what that object was without looking at it. There are other areas that are mapped on the brain called Wernicke's area and Broca's area. And I talked about this a little bit about memory issues and mood issues in our chapter 53. But you can see the area for Wernicke's area shown here as number 22, surrounded by the dashed line on the brain that's shown to the right. And Broca's area is defined, it's the red region of the brain that's surrounded by the dashes. And I told you on Blackboard that I'd give you a picture of this. Now, a scientist named Brodmann, he actually was mapping all parts of the brain, trying to figure out what these parts did. And again, this was the argument for biologic psychiatry theories, right? So I just wanted to link that with a previous lecture that I had posted on Blackboard. Okay, so it's equally important to review what a dermatome is. A dermatome is a specific segment of skin supplied by a single spinal nerve. And again, we've got 31 spinal nerves. So, what's the simplest way to think of this? Let's say you have a pain receptor, and we're gonna poke the anterior side of your knee. I'm going to take a little tack and I'm going to poke the skin just over your knee. Now, do you think the spinal nerve up in your cervical region, C5, is going to relay this information up to the brain about what's happening to your knee? No, it's not. So, it stands to reason that a certain region of the body will be served by the spinal nerve that's closest to that region. And don't get me wrong, we have some overlap as well. All spinal nerves except C1 innervate a segment of skin, and so each of these nerves is associated with a dermatome. Thus, the skin of the body may be divided into sensory segments that collectively make up a dermatome map, which is what I'm showing you here. For example, the horizontal segment of skin around the umbilicus, the navel region, is supplied by the anterior ramus of the T10 spinal nerve. The dermatome map follows a segmental pattern along the body. Dermatomes are clinically important because they can indicate potential damage to one or more spinal nerves. For example, if a patient experiences anesthesia or numbness along the medial side of the arm and forearm, C8 spinal nerve is probably the one that's been damaged. Dermatomes are also involved in referred visceral pain. It's a phenomenon in which pain or discomfort from one organ is mistakenly referred to a dermatome, meaning where it shouldn't be referred to. For example, the appendix is innervated by axons from T10 regions of the spinal cord. Just so you know, many of our internal organs, when they developed, when we were an embryo, they actually developed more superiorly, and then as we continue to develop, they take more inferior positions. As such, any innervation of those organs is probably going to come from a spinal nerve that's higher than what you would predict based on the placement of where that organ currently is. So that's the reason why when someone has appendicitis, they will feel more pain up towards the abdominopelvic region, in the umbilicus region, rather than the abdominopelvic region where the appendix is found itself. Now I can use another example. Heart. When the heart is experiencing a heart attack and there's visceral pain, where does a person usually feel the pain? In their jaw, in their neck, right? In the left shoulder. And that's because embryonically, our heart actually first started to develop in what's now our neck region. And so that's part of the reason why we get the referred pain up in the neck when someone's having a heart attack. Okay, so I'm going to briefly tell you the 3 types of sensory pathways. And again, for our lecture today, I'm only going to focus on 2 of these 3 sensory pathways. Again, sensory pathways are ascending pathways that conduct information about limb position and the sensations of touch, temperature, pressure, and pain up to the brain. Somatosensory pathways process stimuli received from receptors from the skin, receptors from the muscles, and joints. That's what we're talking about. Your conscious awareness of the pain that's happening to your limbs, your skin, and the placement of your body. Obviously, if you were to take your arm and stretch it over your head, eventually you're going to stretch that joint, that shoulder joint, so much that the stretch will then become a painful stimulus. So again, that's what I was talking about, those the pattern theory where some receptors are meant to detect a certain stimulus, but if the stimulus becomes so great, it can then jump over the tracks to, this is now pain. The multiple types of body sensations detected by the somatosensory system are grouped into 3 spinal cord pathways, each with a different brain destination. Discriminative touch permits us to describe textures, shapes of unseen objects, and includes pressure, touch, and vibration. Temperature and pain allows us to detect those sensations, as well as the sensation of an itch. Now that's going to be my primary focus today, that second pathway. And that second pathway is the anterolateral pathway. Proprioception allows us to detect the position of joints, stretch in muscles, and tension in tendons. That's the spinocerebellar pathway, and that's one I'm not going to talk about today. Now, let me then continue on with talking about the anatomy of these pathways. Now, for the 2 pathways I'm going to talk about the most I'm going to discuss the posterior funiculus medial lemniscal pathway, which is a lot to say. This pathway, let's just suffice it to say, is your discriminative touch. Helps us detect textures and shapes. It also helps us to determine pressure and vibration, okay? The other pathway I'm going to discuss is the anterolateral pathway, which is our pain and temperature and itch pathway. Now, what are the similarities for both these types of pathways I'm going to tell you about? They both are going to use a 3-neuron chain. The first neuron is called the first-ordered neuron. Second neuron, second-ordered neuron. Third neuron, well, you get the idea. Third-ordered neuron. Not too hard, right? Now, the first-ordered neuron will always begin out in the periphery of the body. The dendrites of this sensory neuron are part of the receptor that detects a specific stimulus. The cell bodies of primary neurons for both these pathways reside in the dorsal root ganglion of spinal nerves, which is what I told you already. The axon of the primary neuron, or first-order neuron, projects to a secondary neuron, which is found in the central nervous system. That second neuron is called the secondary neuron, or second order. And it is totally contained in the central nervous system, and it's also called an interneuron. Why is it called an interneuron? Well, it's going to connect the first-ordered with the third-order. It's in between the two. The cell body of the second-order neuron resides within the dorsal gray horn of the spinal cord, and the axon of the secondary neuron is going to project either to the thalamus for conscious sensations, or it can go to the cerebellum for unconscious proprioception. But I'm not talking about the cerebellum. So for our two pathways, we're going to be talking about the thalamus, and in a special case, we're going to talk about the reticular activating system. I'll get there. The tertiary neuron, or third-order neuron, is an interneuron. Again, it's totally contained in the central nervous system. And this tertiary neuron, its cell body will be in the thalamus, and then it's going to terminate or end on the primary somatosensory cortex. So again, now let's focus on the posterior funiculus medial lemniscal pathway, and this is for discriminatory touch, pressure, and also for touch, pressure, and vibration, and determining texture of unseen objects. Now just look at this picture here. We see the primary neuron shown in a purple color. You see the neuronal cell body in the dorsal root ganglion. So again, follow this little purple neuron, relaying information about touch, pressure, vibration from the neck, trunk, and limbs. It's going to stay ipsilaterally. See, it's going to stay on the same side that it originated, and it's going to go all the way up to the medulla oblongata. Follow the little purple axon upward. And there it's going to terminate on the second-ordered neuron. And the cell body of the second-ordered neuron is found in the medulla oblongata. And notice the axon of that second-ordered neuron in blue crosses over to the opposite side of the spinal cord. This is where we get decussation. So in the posterior funiculus medial lemniscal pathway, decussation or crossover happens up high in the medulla oblongata. And then that second-ordered neuron is going to terminate on third-ordered cell bodies found in the thalamus, and the third-order axon will terminate on the primary somatosensory cortex on the opposite side of the brain from where the stimulus first initiated these action potentials. So again, we have contralateral perception. The left side of the brain is going to interpret any stimuli from the right side of the body. Now, what are these weird names called fasciculus cuneatus and fasciculus gracilis? Oh boy. Well, the cuneatus, okay, first of all, don't let these technical terms really throw you for a loop. The posterior funiculus medial lemniscal pathway uses a chain of 3 neurons, let's keep that simple, to signal the brain about a specific stimulus: touch, pressure, vibration. Axons of the primary neurons traveling in spinal nerves reach the CNS through the dorsal roots. Upon entering the spinal cord, these axons ascend a specific posterior funiculus. Now, either the fasciculus cuneatus or the fasciculus gracilis. And that's shown here on this picture. And basically what it means is it's a column of axons going up to the brain. The fasciculus cuneatus is axons from sensory neurons originating in the upper limbs, superior trunk, and neck. We're talking upper body. Fasciculus cuneatus, a group of axons coming from the upper body, going up to the thalamus. Okay, no problem. Fasciculus gracilis. Now, where's your gracilis muscle? You think of your lower leg, not your lower leg, you think of your leg, your lower part of your body. Well, fasciculus gracilis tract is carrying axons from the secondary sensory neurons. Sorry, not secondary sensory neurons, primary neuron, the first order neuron, and it's carrying the information from the lower part of your body, like I said, your lower limbs and inferior trunk. So, the sensory input into both the posterior funiculi is organized somatotopically. That is, there is a correspondence between a receptor's location in a certain body part and a particular location in the central nervous system. So let me put that in English. Again, if you look down at the bottom picture here, you're seeing tracts, regions of the spinal cord with axons that would go up, and they're colored either orange or green. The green is going to be, as shown here, the fasciculus gracilis, and the orange is the fasciculus cuneatus. Those are all found in the posterior tracts. And that's, a tract is a fancy word of saying a tract is a funiculus. So, these are axons on the back side of the spinal cord carrying information up to the medulla oblongata, where they will then synapse with the second order neuron, and the second order neuron's axon will now cross over at the medulla oblongata. Now, I am summarizing for you the posterior funiculus medial lemniscal pathway. What is it used for? This is what you should remember. It's used for discriminatory touch, shape of an object, texture of an object, pressure, vibration. Great. We have tracts that are ascending up to the brain that are traveling on the back side of the spinal cord. Great. They are further subdivided into smaller tracts called fasciculi. The tracts that are carrying information about the upper limb, neck, upper body, those are called the fasciculus cuneatus, and the ones that are carrying information from the lower body is fasciculus gracilis. Now, fasciculus is just a smaller collection of axons. That's basically what we're talking about. And we are talking about the first ordered neuron. It's sending its axons up to the region of the medulla oblongata and staying ipsilaterally. Then it will synapse on the second ordered neuron, and the second ordered neuron axon will cross over at the level of the medulla. That's where decussation occurs. Now, when I continue with our second segment, I'm going to discuss the pain and temperature pathways. This is the anterolateral pathway. How is it similar to the one I just showed you for discriminatory touch? One, both pathways use 3 neurons in an order, in a chain. So we're going to still see a first order neuron, second order neuron, third order neuron. How are they different? Where decussation occurs is going to be different in the anterolateral pathway. Decussation is going to be much, much more immediate at the level of the spinal cord where the stimulus occurred, not higher up in the brainstem at the medulla oblongata. Where they get routed for the tertiary or third order neuron can be slightly different as well. And I will explain in our next segment.